Good evening, children. During our last lecture, we discussed about the comparative table between different types of blood corpuscles, the RBCs, the basic characteristics and functions, WBCs, various types, characteristics and the functions, and the platelets. We also started the blood group and blood transfusion, where we discussed various types of human blood groups, why are they called so, and what is a clumping reaction or agglutination in a faulty transfusion of blood. I discussed last time one case in which the person who is the recipient of transfusion is Rh negative and the donor is Rh positive, and what happens as a result of the same. That is the first condition, the first transfusion. In this case, there was an immunogenic response and anti-Rh antibodies developed into the recipient's blood, right? But if a second transfusion is made with same type of Rh positive blood to an Rh negative recipient, this causes clumping of donor's blood, right? And that can be fatal. So today we'll close this topic with another situation, that is Rh incompatibility during pregnancy. Now in this case, what happens is that during pregnancy, the Rh positive fetus, this orange color depicts the Rh positive fetus or a baby inside the mother's womb, that is Rh negative. Now this can be many times possible that the mother's blood is Rh negative and the child in the womb is Rh positive, right? So what will happen in this case? This type of pregnancy will stimulate the formation of antibodies in the mother's blood, because the antigen that is present here, the rhesus antigen, will stimulate the production of antibodies in the mother's blood. Then what happens? In the first pregnancy, enough antibodies are not produced to harm the fetus. That is, the first pregnancy, yes, the antibodies would be made in mother's blood, but they will be really insufficient to harm this fetus. But in case of a second pregnancy, if it is again Rh positive, then more antibodies are added to the mother's blood and a large number of RBCs of the fetus are destroyed. Why are they destroyed? Due to the antibodies formed, and this is due to clumping, which is called erythroblastosis fetalis or hemolytic jaundice or anemia in the newborn child. And this causes extreme illness in the newborn baby, because lack of oxygen also causes the brain damage and other organs, very important, which are for life, they are also damaged. So what I mean to say here, that incompatibility during a pregnancy, in this case of the child that is Rh positive in the womb that is Rh negative, first pregnancy will be alright. But yes, the antibodies have developed into the mother's blood. What if they are insufficient? But if there is a second pregnancy, again of a similar kind, with Rh positive blood of the child, then the child will have a number of problems which can be even fatal, and the child can die also. So this is what we call as erythroblastosis fetalis, and we also have terms like hemolytic jaundice or anemia. I hope you've understood, children. With this, we close our previous topic about blood groups and blood transfusion. Today, we'll continue. Yes, but before we continue with this new topic, let's find out a solution to this. If in a case like this, suppose the father's blood group, let's say, is Rh positive and the mother's blood group is Rh negative. So there is a probability that the child will be Rh positive. So in this type of union of the couple, the mother is injected with RhoGAM or Rh immunoglobulin, which is given in all pregnancies, right? So we call it as Rh immunoglobulin, or we also call it as RhoGAM, and this is injected in the mother's blood so as to avoid this type of erythroblastosis fetalis. Alright, so this can be possible in only in unaware people, but because nowadays technical advancement is so much, that this can be controlled with awareness. All right. Moving further to our new topic, carbon monoxide poisoning. Carbon monoxide poisoning. Let us talk about hemoglobin of the RBCs, which has a great affinity for oxygen. We know it. Where is hemoglobin present, children? Last time we saw it. It's a respiratory pigment present on the surface of RBCs, heme plus globin. Now this hemoglobin has a lot of affinity, that means a likeness towards oxygen, but around 250 times it has more affinity for carbon dioxide, the deadly gas. And when it mixes with carbon monoxide, right, not carbon dioxide, the hemoglobin has 250 times more affinity than oxygen with carbon monoxide, the very deadly gas, and it forms a very stable compound called carboxyhemoglobin, HbCO, right? Now whenever this is formed, this compound cuts down the available oxygen, right? Why? Because the hemoglobin surface that is free is no longer available for oxygen, right? Because the surface is occupied in conjugating with the gas CO. In this case, what happens that there is an acute shortage of oxygen, which leads to unconsciousness and also death. So this type of case happens usually in winters with coke stove burning in a room with all windows closed or in a very limited supply of oxygen. And it's a very common news in the newspapers or headlines that 7 in a family or 10 in a family, they died because of carbon monoxide poisoning. So what is the solution to this? That there should be some ventilation to the room, or oxygen supply should be unlimited to avoid this poisoning. Moving further, there's another thing what we call as the graveyard of RBCs, that is the lymphatic organ spleen, right? Why is it called? Let us justify this heading what we have here, that spleen is called the graveyard of RBCs. The answer is that in a normal male or an adult, here we have around 2 to 2.5 million RBCs destroyed per minute. All right. What is the total number of RBCs? Let's go back in our comparative table. RBCs, they are around 5 to 5.5 million per microliter in an adult male, right? So how much is being destroyed, children? Around 1%. Here, see, 2 to 2.5 million RBCs are destroyed per minute, or around 2 lakh million RBCs in one day. This counts to around 1% of total RBCs that are destroyed every day. And thus, this is why we call as spleen is a place where they are destroyed. So this is why they are called, the organ is called as the graveyard of the RBCs. Another new topic, children, where we're going to start the clotting of blood, also called as the coagulation of blood. Now, whenever there's an injury or a cut to our skin, blood starts oozing out, and the clotting process should begin within 3 to 5 minutes in a healthy body. Otherwise, there will be a severe loss of blood that can cause major complications. So how does this blood begin to clot? What are the stages or steps? Let us see the definitions first. Thrombus or clot. What it is? A clot can be defined as a mesh of fine fibrils of protein fibrin. This protein is called as fibrin, with blood corpuscles and platelets trapped in. So technically, children, what is a blood clot? That there are some insoluble fibers of a protein, and we call them as fibrin. Into the fibers are trapped various blood cells like various blood corpuscles and platelets, and this is what actually becomes a clot that is visible as a hard surface, and that is seen within 3 to 4 days. But the process begins within 3 to 5 minutes. All right. Serum. Straw-colored fluid left after clotting of blood is called serum, and serum cannot coagulate as it lacks fibrinogen, right? So what is serum, children? It is the portion of blood from which fibrinogen has been removed, and because fibrinogen only is the basic of protein that forms the fibers, so serum cannot clot at all. Why? Because it lacks fibrinogen. This you have in class 10, children. So I hope you are understanding. This whole topic of clotting is there, right? But here you'll find, yes, certain things extra. What are clotting factors? Here you will begin with new things. Clotting factors is a term that we use for several proteins present in plasma for clotting, right? So factors means they are nothing but various proteins of the plasma. Plasma means the liquid portion of the blood. We have done it last time. All right. Where it is, just remind you again. This is, I told you, blood is made up of 55 to 60% of that yellow watery fluid called plasma. So this contains proteins, what we call as clotting factors. All right. Let us begin with the process of blood clotting. And this we call it as a cascade of various reactions. Why we call it so? Because in this case, actually one of the molecule when it is formed leads to the formation of others. See, normally these reactions will not happen in a body where there is no injury or a loss to any cell or tissue. But these happen only in case of ruptured tissues or at the site of injury. So why we call cascade of events or reactions? Because formation of one substance promotes the formation of the second substance in another reaction, which promotes the third, and so on. So that is why we call it as a cascade of various reactions. So let's see what is the step 1. In step 1, that is for the process of clotting, let us see it is formation of prothrombokinase or prothrombin activator. Now what happens in this? Wherever we are cut or injured, the rupture of tissue or platelets, they come in contact with the collagen fibers which disintegrate, and now they release a factor 3 or TF. We also call it as the tissue factor or tissue thromboplastin. And the composition of this is phospholipids, lipoproteins and proteolytic enzymes. Proteolytic means lysis of protein substances, and this is all that is new for you, children. Yes, the step 1 is there, that the substance thromboplastin is formed. How is it formed? From the ruptured tissues or the platelets that are destroyed, and this factor is formed. But composition is totally new, children. Please learn it properly. So there are phospholipids, lipoproteins and proteolytic enzyme. Alright. Now, what happens further with this? This substance further in combination with clotting factor 7, right? Calcium ions and some more factors like 10, 4 and 5, right? They convert into prothrombin activator complex or prothrombokinase complex, right? And one more thing I'll add up here, that the clotting factor 7 is called as serum prothrombin conversion accelerator, because this will speed up the conversion of this complex or formation of this complex. So step 1 is over. Once again, before we come to step 2, in this step what has happened? First, we form the substance tissue thromboplastin from the injured or ruptured tissues or platelets, and then further with many factors 7, 10, 4 and 5. We always write them in Roman numbers. We have formed a complex called as the prothrombokinase complex. Now, this thrombin, that is thromboplastin, that is formed here is an inactive form, right?